So thank you, Dr. Overdoing, for inviting me, and thank you everyone for waiting till the last talk. I have a I'm a computational person, so this story will be a little different from all the impressive talks we have heard so far. So I'm Rituparna, and thank you for the introduction, Manan. I'm Rituparna. I'm an assistant professor at the University of South Florida. Um, I have a super young lab, about a year old. So the story I'll be sharing will mostly be from my last days at Professor Gray's lab as a postdoc. Uh, all right. So my lab does computation on different sets of problems. We look at both, so we try to answer questions around how do proteins behave in different environments, particularly in the cellular membrane, and proteins behaving in presence of polymers. So we look at dimerization of receptor tyrosine kinases, looking at self-assembly of proteins and polymers and their phase behaviors, also some biomaterial applications. Um, yeah, but today's talk, I'll be talking about methods development for understanding where proteins or membrane proteins form an interface. I think I don't need to give you a big introduction of how membrane protein complexes are important. So they obviously, you all know that they play an important and a fundamental role in regulating cellular function. So here I show glucose transporters, conformational change in myosin proteins, or like we just heard from the previous speaker about receptor tyrosine kinases. And so they are great, but they are a difficult system to handle. So despite their significance, modeling membrane-specific protein-protein interactions are significantly challenging, and I'll tell you a few reasons why. Particularly, the environment is extremely variable. So here I show the head group composition of mammalian plasma membrane. So and that kind of leads to my second point, where the number of structures of membrane proteins are relatively small. Um, so, um, so we think that the computational tools can help discover more about membrane proteins here. But if we come to the computational side of it, the whole complexity of so many lipid types, the solvent, salt, and the big proteins makes the system pretty complex and hard to compute. So here, for example, I'm showing Jabberdoc, which is a molecular dynamics based simulation where they first equilibrate the membrane in the membrane environment and then extract their volumetric mapping. This works great. This is and takes about 4 days, many GPUs to calculate where two proteins forms an interface. So during my postdoc, we were thinking about these systems, and we thought that if a system takes so much long time, it it definitely forms a difficulty when it comes to like docking larger proteins, or basically forming or looking at long length scale problems. And if we can think of designing these membrane proteins where you have to change the residues and then figure out whether the interface is binding stronger or smaller, then that's again a problem which is intractable at that point. So during my postdoc, I co-developed this method, which we called implicit membrane models, where what we did is we replaced the lipid and the solvent system as a continuum model. So we modeled the lipids separately and took the properties of that, and then on top of that, we replaced an all-atom protein. So if relative to coarse grain system, which kind of groups multiple atoms. Um, it's still sometimes intractable, but then because we have now a continuum system replacing the lipid and the solvents, we could make those calculations about 50 to 100 times faster. So because we could save that time, now we are at a position to look at much longer scale, longer length scale systems. We can also do design, and those are some of the examples which I'll be showing. So in today's talk, I'll be talking about an application of the system where we are talking about docking to proteins, particularly transmembrane proteins. So we developed this on the Rosetta protein design suite, where Rosetta is like, this is a framework to study protein. We are interested in equilibrium structures always. So we chose something called a computational method called Monte Carlo, where we take the structure of protein and we perturb the sampling space, meaning if we want to see where two proteins are docking, our sampling space is position of the proteins. If we want to say design new proteins, our sampling space is residue of the protein. So depending on your application, we perturb the sample space. We then calculate the energy with a formula which we call score function. And then we do it iteratively till we reach the lowest energy state. However, the biggest challenge always has been, are we sampling enough, or are our scoring methods good enough to represent what is in nature? So the idea or the approach we have taken is an iterative method of developing applications, developing score functions, and then trying to always benchmark it with experimental evidences, then go to our collaborators, do some applications, and again come back and improve our models. So, yeah, so that's the approach we have taken. So that's something which I'll be discussing today as well. We built a model for docking. We will benchmark it on available data sets, and then we'll go ahead and do some applications with our collaborators. So I talked about the scarcity of structures as one of the difficulties, and the diversity of the environment as the few challenges. But then there are other challenges where I'm talking about flexible protein backbone. So our method handles flexibility in the protein backbone. Okay. So to show the difficulty of the problem, here I'm bringing in soluble protein complexes. So here I'm in gray, I'm showing bound soluble protein complex, and on blue are the unbound complexes. The difference in the unbound and the bound complex is shows how flexible. The greater the difference is, higher the flexibility it is. And here on your right, I'm showing a result from a Capri challenge where they are trying to dock. So different methods are compared to dock some blind challenges. And x-axis, there are different methods. On y-axis, it's a score of how good the prediction is. Higher the number, better it is. So on the x-axis, they are also like um, they're also arranged in the order of flexibility. So on your right extreme, it's the most flexible targets. And on the left, you have the rigid targets where the difference between bound and unbound structures are lower. So you can see as the rigidity of the protein backbone increases, we have a range of methods which are good. But as the flexibility of proteins increases, there are not many methods which can predict them better. So this is kind of showing you that there is a lot of challenge in particular predicting predicting docking interface for flexible targets. The way we circumvent the challenge here is we take two proteins and develop ensembles or add flexibility to the backbone of these monomers, and then we do pre-packing of the side chains and then do sampling of the structures. So it's not only sampling of their backbone, it's not only sampling of the individual positions, it's also sampling of the backbone of individual proteins. So we have a rigid docking protocol where we don't change the structure of the backbone, and we have a flexible protocol where we change the backbone of the um, uh, proteins. So here I show you first two targets. One is an extremely rigid target, one COY, where we have the answers already. The gray is the bound complex, and the blue and the green are overlaid on top of the bound complex. They are unbound monomers. Green one is one monomer, blue one is another. And here on your right, I show a flexible target where again, it's the same color. So the gray is the natives bound complex, and the green and the blue are native unbound complexes. What we do is we take the unbound complexes and try to see where the interfaces are. And the way we do it is even though we know the answer, we separate them and then try to find out where they face. So to do that, so on this plot, every dot is a structure. So every dot is a prediction of two proteins coming together. And then we calculate the energy of the structures shown here on the y-axis, and how far it is from the answer, or how far it is from the native bound complex is on the x-axis. It's the root mean square deviation from the bound native complex. So as you can understand, if we have a very low score, it's good. If we have a very small difference from the native structure, it's good. So we would want our predictions to lie on the left corner of this plot. So here on one column, I have the rigid target. On the right column, I have the flexible target. So first we use our rigid docking protocol, and we see that for our rigid protein structure, rigid bound complex, we have a very nice prediction. It's right at the bottom corner here, and we have very good predictions. However, for the flexible target, we don't have decoys which are closer to the native bound complex. So then we used our new protocol where we added flexibility to the backbone. And then here, like our rigid case still has good answer, but for the flexible one, we now have much better answers than the rigid protein docking. So we not only did it for one structure, in total, we collected a benchmark of 29 transmembrane protein complexes for which we have both a bound complex and an unbound monomer. So here we compare the interface root mean square deviation from the native bound structure of the rigid Rosetta MP dock and compare them with ensemble docking, the new protocol. For all cases, we also compare it with with the previous method, Jabadoc. So here on the right, you have the highly flexible targets, and on the left, you have medium flexible targets. So for all the proteins, we see the rigid dock uh, so the ensemble docking, so adding flexibility to the backbone, has improved the predictions for most of the cases, and has done overwhelmingly better than the previous method, Jabadoc. And this is faster. So while we were developing this method, AlphaFold Multimer came, which is a deep learning based method where the input is the sequence of monomers and the output is this 3D structure of the complexes. So here I'm showing you an example. So here again, gray is the bound complex, overlaid on top in color are the monomers. Here, this is the AlphaFold multimer prediction, where if you, so this is the top view and the side view, and you can see from the, particularly from the top view, that the interface is not correctly predicted by AlphaFold. So what we did after that is we took this prediction from AlphaFold, ran into our method, and we could correct it. So we again, the question which we are trying to answer is, could we take predictions from AlphaFold and refine through our method? And so here, these are the predictions from AlphaFold multimer shown here in the gray bar. And when we refined all of these structures through the ensemble docking method, we are much better than both the AlphaFold prediction as well as the Jabberdock. And in our only, so this is the hybrid method. So the hybrid method definitely does better than all other methods. So here it's a bit more clear from here. If we have interface RMSD on the y-axis and the interface RMSD of AlphaFold predictions only, for all the cases, the predictions are made better through the Rosetta MP-Doc method. So it could reduce the, it could correct the interface shifts from AlphaFold. So so when I started the lab, then I'm still continuing to work in this problem. And then AlphaFold 3 was released. So, and then obviously the question was, can AlphaFold 3 predict or capture the membrane associated protein docking? So here is an example of prediction from AlphaFold 3. This is from AlphaFold multimer. And then with AlphaFold 3 here, the teal is the best model which AlphaFold 3 gives, and pink are all the other methods. Gray is the native structure. So if you can see, all the predictions are far away from the native structure. And when we used AlphaFold multimer plus Rosetta MP-Doc, we could correct that. So we continued this. So for all the benchmark proteins here, we ran AlphaFold 3 for several times with different seed values. And this is our result for all the 29 proteins. The prediction from AlphaFold 3 on our benchmark varies from somewhere. So the y-axis is now root mean square deviation from the native complex structure on a log scale. So we have sub-angstrom level precision to somewhere for a group of proteins which the difference is pretty high, much more than 10 angstrom. We are continuing to work on this to answer questions like what are the chemical signatures of these interfaces? Can we use AlphaFold 3 predictions for analyzing these interfaces much better? So to summarize, I have shown you Rosetta MP-Doc today, which samples large conformational ensembles of flexible monomers and docks them together, transmembrane protein targets, using an implicit membrane environment. Using the implicit membrane environment makes it faster, so we can handle larger proteins. However, so we could show that Rosetta MP-Doc successfully predicts the correct interface with 67% moderately flexible targets and 60% of the highly flexible targets. However, we are not happy with the 67% and 60%. We still fail to capture large conformational changes. So my lab is taking the work forward where we are adding more flexibility. We are adding definitely different. So far, we have just scratched the surface with the implicit membrane model. We have really, like, we can add membrane curvature. We have just done homolipid layers, so we can do mixed lipid layer characteristics and like a lot of other properties which we can keep adding. Uh, so our protocols are open source. If you download Rosetta, our protocols are there. We are in the process, the scripts are available on GitHub, and we are in the process of making it as a web server. The work was majorly done in Professor Gray's lab with a couple of my co-authors, Dr. Harmalkar, who is a graduate student at Jeff's lab, and an undergraduate, Priya. Our code reviewers were Dr. Woods and Dr. Harmalka. Again, special thanks to the funding agencies, computing resources. We have now, in my lab, I have a couple of my students who are taking this work forward, particularly analyzing deeply how the AlphaFold 3 predictions are working. We are also working in collaborations with Dr. Pine's lab, where we are looking at dimerization of proteins in the inner membrane of inner mitochondrial membranes. So with this, I would like to thank you all for your attention. I would love to take a lot of questions, comments, and learn a lot from you all.
Hey, thank you very much for the talk. That was a wonderful talk. With that, I see there's questions from Michael, but if anybody else has questions, feel free to ask them in the chat or just unmute and ask your question, but we'll start with Michael.
Okay, well, anyone else is welcome to go first. I asked too many questions, but a fascinating talk. So I have a couple. I'm just wondering if you've ever tried a beta barrel structure refinement with MP-Doc.
We did not, but beta barrel is a protein of interest for us. We are doing some design applications for beta barrel, but I would love to take it as a challenge for docking as well.
Okay, great. And can you comment a little bit more about how you accommodate different lipid bilayer properties like the charge or the stereochemistry of head groups, possibly, maybe even bound metals, calcium, or whatever, and the asymmetry of the bilayer? Can that be accommodated with your MP-Doc program?
Yes. So far we have a very simple uh, homolipid membrane. So we have the upper layer and the so we don't have any difference in the lipid types. But we have a library of 13 different lipid types, ranging from different chain length and different charge groups. We accommodate the electrostatics interaction with the head group by we do an all atom simulation for our lipid groups and calculate the electrostatic field. And then when we have an all atom protein, we have the interactions with that electrostatic field. We also incorporate difference in dielectric constant in the lipid layer versus outside the membrane. And then we have hydrophobicity calculations based on the water density and from experimental evidences of how a residue, the delta G difference of a residue from outside the membrane and inside the membrane.
Thank you.
Yeah, but we can we can include different kinds of lipids or cholesterol or other varying types of lipid properties.
We can't hear you, Monan, but Rashish, maybe you can unmute and ask your question.
Yeah, it's just not a scientific question, but how do you manage to get funding in this dynamic field when you're competing against DeepMind, Amazon, Embol ABI?
Yeah, that's a good question. I'm young enough, so I'm trying hard. But I mean, that's why when I was looking for jobs, I chose to stay in membrane proteins because honestly, DeepMind and all the AlphaFold trainings are more for soluble proteins. So we work really hard to always benchmark the new tools on the tiny little benchmark sets we have. And so far, we have seen, okay, our jobs are safe, but yeah, it's hard.
Thank you for the answer there. We also have a question from Scott. Scott, would you like to unmute yourself?
Sure. Yeah, no, I was just curious. I was sort of inspired by an earlier talk that we saw on COVID, right? And I was just wondering, when you using these algorithms, what have you ever tried small viral peptides that are meant to aggregate and insert? Like, what happens with this sort of simplified continuum dielectric model that you're using?
We didn't. I just wrote that in my copy that, okay, these are good peptides and protein models to test.
Uh-huh. So you've done it, or?
No, we haven't. We
Okay,
will
cool.
be
Right.
doing it. Yes.
Okay, one more question just about the Rosetta, just like with Rajesh's question, is there funding for the continuation of the Rosetta community? And I know that was a topic a year or two ago. Is that moving ahead? Does that help you with funding, or the other Rosetta researchers in the space?
So we have small internal fundings, but that's, I mean, we all the Rosetta community depend on federal funding and and collaboration with industries for funding.
So if pharma want to support the Rosetta or the MP Doc project, how do they support you? Do they just get in touch with?
Yes, they get in touch with the developers, and then we take it from there.